Hello everyone, this is Mark Dugay, a graduate student of Grand Canyon University. In this presentation, an aspect of the urinary system, particularly in the fluid compartments of the body, including major ionic composition, will be explained. Water is the most abundant component of the body. As shown in the figure, the water content of the body's organs and tissues varies from as little as 8% in the teeth to as much as 85% in the brain. Human beings are mostly water, ranging from about 75% of body mass in infants to about 50 to 60% in adult individuals, to as low as 45% in old age. The percent of body water changes with development because the proportions of the body given over to each organ and to muscles, fat, bone, and other tissues change from infancy to adulthood. Our brain and kidneys have the highest proportions of water, which composes 80 to 85% of their masses. In contrast, teeth have the lowest proportion of water at 8 to 10%. The water content of the human body is being maintained and regulated by the kidneys. However, the percentage of water in the body varies from person to person due to the variability in the amount of adipose tissue, or commonly known as fat, whereby the adipose tissue has a low water percentage compared to other tissues. Blood has 50% water. Even the soft tissues such as skin, muscles, and internal organs such as heart, lungs, liver, and the bones have 20 to 25% water. And the driest fat tissue has only 10% water content. This figure presents the fluid compartments in the human body. These include the intracellular fluid compartment, a system that includes all fluid enclosed in cells by their plasma membranes, and the extracellular fluid compartment, which surrounds all cells in the body and has two primary constituents: the fluid component of the blood known as the plasma or the intravascular fluid, and the interstitial fluid that surrounds all cells not in the blood. These compartments allow material to travel between cells and the plasma in capillaries through the interstitial fluid. There are also two minor compartments included in the extracellular fluid: the lymph and the transcellular fluid. Lymph is the fluid being returned from the interstitial fluid to the plasma by means of the lymphatic system, where it is filtered through lymph nodes for immune defense purposes. Transcellular fluid consists of a number of small, specialized fluid volumes, all of which are secreted by specific cells into a particular body cavity to perform some specialized function. The table shows the major body fluid compartments. The total body fluid represents approximately 60% of the body weight of a person, or 42 liters. It is distributed between the two major fluid compartments. First, we have the intracellular fluid and fluid surrounding the cells, or extracellular fluid. The intracellular fluid compartment consists of about two-thirds, or 40% of the total body weight, or 20 liters, and the remaining one-third, or 20% of the total body weight found in the extracellular fluid compartment is further subdivided into two: plasma and interstitial fluid. Plasma, the fluid portion of the blood, makes up about one-fifth of the extracellular fluid volume, or about 3 liters. Whereas the interstitial fluid, which lies in the spaces between cells and makes exchanges with the cells, represents the other four-fifths of the extracellular fluid, or approximately 11 liters. The figure presents the ionic compositions of the major body fluid compartments. As shown in the figure, the plasma and interstitial fluid are similar in composition. Sodium, potassium, chloride, bicarbonate, separated by a highly permeable capillary wall. Accordingly, plasma and interstitial fluid are nearly identical in composition, except that interstitial fluid lacks plasma proteins. The presence of higher concentration of proteins in the plasma differentiates these two extracellular compartments. Due to the low permeability of capillary membranes to the plasma proteins, only small amounts of proteins are leaked into the interstitial spaces. The extracellular fluid, including the plasma and interstitial fluid, contains large amounts of sodium, chloride ions, and bicarbonate, but only small quantities of potassium for both and protein and plasma. The composition of extracellular fluid is cautiously regulated by various mechanisms, but especially by the kidneys. This regulation allows the cells to remain continually immersed in a fluid that contains the proper concentration of electrolytes and nutrients for optimal cell function. In contrast to the similar composition of plasma and interstitial fluid, the composition of the extracellular fluid and intracellular fluid differ markedly. Each cell is surrounded by a highly selective plasma membrane that allows the passage of certain materials. In contrast to the extracellular fluid, the intracellular fluid contains only small amounts of sodium ions. Instead, it contains large amounts of potassium and phosphate plus protein, which is almost 4 times as much as in the plasma. So these are the ionic compositions of the major body fluid compartments. This scholarly article titled Assessment of Fluid Shifts on Body Compartments Using Both Bioimpedance Analysis and Blood Volume Monitoring is related to the assignment presented. This study considered the body fluid shifts that are commonplace in chronic hemodialysis patients during the dialytic and interdialytic periods and are associated with dialysis-related complications. Hence, in this study, the researchers evaluated fluid shifts of body compartments using both bioimpedance analysis, or the BIA, and the blood volume monitoring, or the BVM, from the start to the end of hemodialysis. Bioimpedance analysis is a method of evaluating the composition of the body, which is specifically used to determine the distribution of body fluid compartments, including the intracellular fluid, extracellular fluid, and the total body water. Whereas blood volume monitoring is essential in estimating the relative changes of the intravascular volume or the blood plasma. All patients underwent low-flux hemodialysis 3 times weekly with a bicarbonate buffer for 4 hours using the Fresenius machine with a built-in ultrasonic blood volume monitor. A multi-frequency bioimpedance analysis analyzer was used to measure resistance or impedance of 6 frequencies ranging from between 1 kilohertz and 1 millihertz. In this study, the experimental group was using both the bioimpedance analysis and blood volume monitoring to evaluate fluid shifts of body compartments compared to the recent studies using only either of these methods, which served as their control. Table 1 depicts the baseline characteristics of the 24 hemodialysis patients with a mean age of 57 years, a 14 men and 10 women, 10 diabetic and 14 non-diabetic. These patients were on hemodialysis therapy for a mean of 64 months, and patients' pre-dialysis and post-dialysis mean blood pressure were measured, plus their body weight, height, and their mean ultrafiltration. The ultrafiltration volume is the amount of water that must be removed in a single treatment to return a patient to his or her target or base. All patients underwent low flux hemodialysis 3 times weekly for 4 hours. A figure 1 shows the time course differences of blood volume, intracellular fluid, extracellular fluid. As shown in the figure, the blood volume reduced significantly from the start to the end of hemodialysis. This relative change of volume was progressively reduced during the first hour to the last hour of evaluation. During the hemodialysis, fluid was removed from both extracellular and intracellular compartments. Extracellular fluid was predominantly reduced compared to intracellular fluid until 3 hours of hemodialysis. After, after 3 hours of hemodialysis session, reduction of intracellular fluid became steep. Mean percent reduction of intracellular fluid was not significantly different to that extracellular fluid at the end of hemodialysis. Figure 2 shows the time course differences of body compartments, the arm, trunk, and leg. As depicted in the figure, the fluid of arm, trunk, and leg is significantly and progressively decreased from the start to the end of hemodialysis. Percent fluid reduction of arms, trunk, and legs were approximately 13%, 9%, and 7%. Based on the figure, the arms had the greatest fluid reduction, but there were no statistical differences. Okay, figure 3 presents the three patterns in reduction of blood volume. A had the type 1, which is the continuous and linear reduction rather. B had the type 2, which is curvilinear reduction, and C had the type 3, which is early reduction and plasma refilling. As presented in the figure, the patients of type 3 early reduction and plasma refilling had higher extracellular fluid and total body weight ratio, and the extracellular fluid and ultrafiltration volume at the end of hemodialysis session compared to the other two types. And the blood pressure was monitored and had the same in all the three types at the end of hemodialysis. Table 2 shows the clinical differences of three characteristic patterns in blood volume change. As shown in the table, type 2 patients had the most hypotensive events among these three groups. The type 3 patients had the higher extracellular fluid and total body weight ratio, extracellular fluid and ultrafiltration, and lower ultrafiltration volume at the end of hemodialysis session compared to those of type 1 and 2. These findings mean that the patients of type 3 are in excess of volume status. Based on the results of the study, the use of blood volume measurement in chronic hemodialysis patients as a tool both in preventing intradialytic hypotension and in identifying patients with volume overload who could benefit from intensified ultrafiltration. Moreover, the changes of the body fluid at the end of the hemodialysis was similarly removed from both extracellular and intracellular compartments, and the percent reduction of arms was the greatest in chronic stable hemodialysis patients. The pattern of blood volume change measured by blood volume monitoring is useful for determining more accurate dry weight and correlating volume status in patients with hemodialysis. Okay, here are my references. Mahalo.